Okay, so we have been talking about the cell. So we have only completed the what? Cell membrane, right? Now I'm going to talk about everything inside the cell. What do we have inside the cell? You have the what? Everything inside the cell is actually the what? The base right here is the gel-like structure. We call it what? Cytosol or cytoplasm, right? Inside the cytosol, you have all these organelle. Like as I told you in the beginning, whenever we are thinking about the cell, we are actually talking about a big mansion with a lot of rooms, right? All these rooms that we have, we call them what? Organelles. Okay, and then inclusion, not only the organelles in the cytoplasm, you also have some molecules submerged in there, and we call them what? Inclusion. Like for example, the molecules like the storage molecule, glycogen granule, pigments, lipid droplets, vacuoles, crystals, these are all inside your cell. So now, as you can see right here, so you guys know that in a mansion or in a house you have so many rooms, right? Do you think all the rooms have the walls in it? Some rooms are open, right? Right, like the kitchen, right? Kitchen is open to the living room a lot of the time, right? And kitchen and living room together are the same kind of the room, right? And then you have the different bedrooms, like you have the bedroom for you, maybe your brother, maybe your sister, maybe your mom and dad, maybe your grandparents, if you have a big joint family. So now your cell is exactly like that. So in different rooms also have different function, right? Maybe your brother room has a big video games, right? And what does your brother do? Like that, right? And maybe in your room you have a big dressing table. What do? Right? And your like grandparents room probably have a large big TV so that they can watch TV, I don't know. And then probably your parents room have something to do some office work or something like that, because a lot of the time probably parents work from home too. And you have the kitchen. What happens in the kitchen? Hmm? What happens in the kitchen? You cook. Very good. And then probably you also have a dining table, right? Where you serve the food. And then probably you also have a living room, right? And then in the whole room, you also have a lot of walls and structure to have the what? Proper structure of the house, right? Exactly like that, your cell works. Exactly like that. Okay. So now let's talk about it right here. First thing I want you to understand that the room of your parents is your what? Mitochondria, because the parents are probably the one who's earning the money, right? The room of your parents is the what? Mitochondria. Why? Because the mitochondria is also called the what? Powerhouse of the cell. Why the mitochondria is called the powerhouse of the cell? Because you are generating energy. What is the name of the energy molecule in your cell? ATP. You are generating ATP in the mitochondria. So these are the mitochondria, your parents' room. They are generating the ATP, and we call it powerhouse of the cell. So if you look at the structure of the mitochondria, you see that they have the outer membrane and the inner membrane, and the inner membrane has a lot of what? Fold to increase the surface area, to increase the energy production. Okay, so the currency, the money, okay, is happening in the mitochondria. Okay, now before I talk about endoplasmic reticulum and the Golgi apparatus, I want to talk about the nucleus. But let me also tell you a little bit about the peroxisome and the lysosome. So the peroxisome is in your house probably the laundry room. Paroxysm, what is the paroxysm doing? They are detoxifying. If you have anything bad in your cell generated, that is something you are trying to detoxify by the what? Paroxysm. Lysosome is also, I could say, in the part of laundry room, because if anything bad is happening, what you are trying to do? You are just recycle it. Okay, so it's the recycle center, I would say, lysosome. So we'll talk more about that. So these are the rooms which has actually the what? Wall around it. So these are the specific rooms, but the some rooms does not have the walls around it. Like for example, the kitchen, right? And in your cell, the kitchen is called the what? Ribosome. What do you call the kitchen? Kitchen is the what? Ribosome. Now, what about the cytoskeleton and the centriole? Cytoskeleton, I told you a little bit about it earlier, which is basically providing the structure of the cell, right? Have you ever seen tent? Tent is basically the what? A big cloth. So now the big cloth on top of the stick, right? What happens to the tent if you do not have the stick? You just have the cloth that does not have any structure. To be able to hold the structure of the tent, you need to have the what? Stick. And those sticks are called the what? Cytoskeleton. And there are different types of the cytoskeleton, and some of the cytoskeleton actually together make the centriole. Okay, so this is basically the overview. So now let me try to make a story out of all these things so that you remember about the function of all of the different organelles. So to be able to talk about the story, the first thing I want to talk about is the nucleus. Nucleus is called the what? Control center of the cell. Do you know why nucleus is called the control center of the cell? Why? Why nucleus is called the control center of the cell? It has DNA. What is very special about the DNA? Huh? Genetic information. So when you say genetic information, what do we actually mean by that? Huh? Able to replicate, reproduce. That's important. But anything else, can you tell me? Huh? How do we look like? Yeah, very good. So let me tell you the thing, which is in your body, like a lot of the function, like for example, what is the color of your eyes? What is the color of your hair? Is it wavy, or how tall are you, or what enzyme you can produce, what protein you can produce? These are all determined by the information that you have in the DNA. Okay, so I would talk about that as a recipe book. So your DNA is basically the what? Recipe book. So for example, in the house that we are talking about, in your house that you have so many rooms, there you have a big recipe book. The recipe book probably is coming from generation to generation. Very important recipe book. Okay, so you have the recipe for different type of the like dishes. Like for example, you have the recipe for taco, you have the recipe for biryani. Do you know what is biryani? Yeah, good. Okay, so you probably have the recipe for pizza, you have the recipe for all different type of the food, like maybe like 25,000 different type of the dishes. The recipe is there in the DNA, the information is there. This is the what? Information. Okay, so now you have the information in the DNA. So now you have the recipe book at house for the 25,000 dishes. But do you cook all the food together? 25,000 dishes? You do not, right? For example, one day probably you want to eat taco, then you go to the kitchen and cook taco, right? And then for example, probably some other day you want to eat pizza, you just look at the recipe of the pizza and make pizza, right? And then for example, if you want to make something, so you see what I'm trying to tell you, right? So similar to that, for example, in your body, do you guys know, is there anyone who's lactose intolerant? Okay, I saw someone is lactose intolerant. So what is the difference? Your name is Liliana, right? So the difference between the Liliana in and all of you, the difference is when you drink milk, you can digest the milk, but Liliana cannot digest the milk. But why? What is the reason for that? The reason is lactose is a disaccharide. So the lactose is what? A disaccharide, which is, remember we talked about it like last week, that lactose is a what? A disaccharide. It has the two sugar. What are those? Glucose and galactose, right? Glucose and galactose. Now you cannot absorb lactose like that. You have to you have to digest it to what? Glucose and galactose to be able to absorb it so that your body can use it, right? Now, as I told you, there is another thing called enzyme, the protein, right? What is the function of that enzyme? To speed up the reaction. This reaction is not possible without the help of the what? Enzyme called lactase. Okay? So now, whenever I drink milk, my nucleus in the DNA, I have the recipe to make the what? Lactase enzyme. So my body will work to make sure I can make the lactase, so that the lactase come and what? Break down the lactose to the glucose and galactose, so that I can use the glucose and galactose. So that is why I am not lactose intolerant. But if you cannot make this lactase enzyme, what? What is going to happen? You cannot digest the milk. So who is actually determining that? It is determined by the recipe book. Do you have the recipe in your DNA, or you do not have the recipe in your DNA? For example, also it could be you have the recipe, but the recipe is not correct. So you are not properly making the proper dish. So that is exactly what is happening in our nucleus. So whatever information we have, whatever, however recipe you have, like for example, I make biryani and you make biryani is probably different. Why that is different? Because your recipe is different than mine, right? Similar to that, my eyes look different and your eyes look different because my DNA has this information and your DNA has a different information. Does it make sense? My skin color and your skin color is different because probably I have a different information for my skin color and you have a different information for your skin color. So do you understand what I am talking about? So why do you think, do you see why the nucleus is called the what? Control center, right? So nucleus has the chromosome, actually the chromatin. Chromatin is actually made up of the DNA and the protein, but we are talking about the DNA which has the gene. The gene is basically the DNA. When I said DNA, DNA is basically the recipe book. So it has the recipe for all the different dishes. But whenever I am saying gene, what I'm talking about? I'm talking about recipe for taco, recipe for pizza, like in your body, recipe for making collagen, recipe for making lactase, recipe for making insulin, right? So these are, for example, do you guys know that the type 1 diabetes, you cannot even make insulin, right? You cannot even make insulin. So probably you cannot make that insulin protein, probably you probably have the problem in the what? DNA. Or maybe there could be another reason too, but this is one reason. So you see what I'm trying to tell you right here. So you are you because of the information that you have inside your DNA. So now you also, not because of the information only, but also what you cook. Do you actually cook it? I might have the recipe book at my home. That doesn't mean that I am cooking all the 25,000 dish every day. I only cook the dish that I want to use right now, right? So it's not only the gene that you have, but also expressing the gene, the cooking the gene, the cooking the food, also making the protein, right? It depends on that. It's almost, I also can think about it like this. So in this room, I have the light, right? But the light is not going to work if I do not turn on the switch. Exactly like that, you have the information. If you want to cook the food, that's when you will have the food. If you do not cook the food, you will not have the food. So similar to that, we have in our cell, we have the 25,000 gene in our cell, and all of the cell has those 25,000 gene. But not all cells actually cook all the different food, right? For example, pancreatic beta cell produces insulin, but you have the insulin gene in your skin cell too, but your skin cell do not produce what? Insulin. So do you see what does it mean? You might have, like for example, in a neighborhood, you have your house probably love to cook taco, but maybe the house by you does not like to cook taco. They want to probably cook biryani. So does it make sense? Do you see what I'm trying to talk about? Okay. So now let's come to the point. So first of all, you have the information to cook the food in the where? In the DNA. So now after you have the information to cook the food in the DNA, what do you want to do? To cook the food, you want to, like for example, now let's actually come to the point. You have the big recipe book. 25,000 recipe in there. How big the book is? Very big, right? So before you cook the food, you need the what? Ingredients. So what if now for the ingredient, what do you have to do? You have to go to the store and buy the ingredient, right? So now let me talk about it like that. For example, today you want to make taco. So the first thing probably you want to do is you open the recipe book, and you cannot take that big recipe book to the store. What do you want to do? You want to copy the information from the recipe book to a piece of paper so that you can bring all the ingredient. So when you are copying the information from the recipe book to a piece of paper, that process is called the transcription. What do we call it? Transcription. So you are copying the information from the DNA to RNA. So that is called the what? Transcription. So now you have that transcript. We also call it messenger RNA. What do we call it? Messenger RNA. So now you go to the store with that messenger RNA and look at all the ingredients and bring all the ingredients. By another type of RNA, we call it transfer RNA. What do we call it? Transfer RNA. So now you went to the store with the messenger RNA, bring all the ingredients by the transfer RNA. After you bring all the ingredients, where do you want to take it to? Kitchen. What is the kitchen of the cell? Ribosome. So you are going to take the ingredients to the what? Ribosome. So in the nucleus, you have your transcription happening. So you copied the information from DNA to RNA. Now RNA has to come out of the nucleus and bring all the ingredients to the what? Ribosome. So we have the two different type of the ribosome that are attached to the endoplasmic reticulum to give the appearance of the rough endoplasmic reticulum. You also can have the free ribosome. Okay, and these ribosomes are called the what? Kitchen. So you bring all the ingredients and take it to the what? Kitchen to cook the food. So the food, the making the protein, the synthesis of protein is happening inside the what? Kitchen. Okay, now you cooked the taco inside the ribosome, rough endoplasmic reticulum. On top of the rough endoplasmic reticulum, you have this ribosome. In this ribosome, you are cooking the what? Protein. After you cook the protein, so for example, now what did you do? Now in your home, you cooked the taco. After you cook the taco, before when you take it to the dining table, before you take it to the dining table, you do some modification, right? You do some modification. Probably you put them in a dish, and then probably you put some cheese on top, cilantro on top, whatever, right? So you are doing some, making some changes after making the what? Protein. And that is happening in the what? Rough endoplasmic reticulum. So in the kitchen, which is the ribosome and the rough endoplasmic reticulum together is actually helping with the what? Synthesis of the protein and the modification of the protein. So now after you modify the protein, what do you want to do? You want to send the food to everyone. Like for example, you have 5 people, maybe 7 people at home. So you have your brother in brother's room, your dad in dad's room, your everyone is in everyone's room, right? So now you want everyone to come to the what? Dining table. But your brother is in the middle of the what? Game. Your father is probably in the middle of the what? Work. So everyone is, maybe your grandparents are in the middle of the movie, so they cannot come to the dining table. They want to eat where? Inside the what? Inside the room. Now before you take it inside the room, so do you think everyone at home eat the same way, the same food? No. What happens? Probably you do not like cilantro. So what do you have to do? We have to take out the cilantro from there, right? Make your plate. And then your brother's plate, your brother probably does not like cheese, so you take out the cheese from there. And then probably your father has a high cholesterol, does not want the meat, so you take out the meat from there. And your grandparents probably cannot eat meat, and also probably they want some other type of the, maybe they want you to add some avocado in there. So what is happening? So first of all, you cooked it, you had some preliminary modification, and then before you actually send it to everyone, you had to do more modification based on their own criteria, right? So now from the endoplasmic reticulum, the dining table is the what? Golgi apparatus. So you are sending the protein from the endoplasmic reticulum to the Golgi apparatus. Just to let you know, here whenever you are moving from the ribosome and endoplasmic reticulum to the Golgi apparatus, they cannot just go like that. You have to actually load them in the vesicle, and with the vesicle you are sending to the Golgi apparatus, and these vesicles are called the transport vesicle. Remember I told you about the what? I told you about the car, the vesicle. So these are the what? Transport vesicle, which is carrying the protein from the what? Endoplasmic reticulum to the Golgi apparatus. So now in the Golgi apparatus, you are making all the modification. First of all, what you are doing? You are sorting which one is going to go to your room, which one is going to go to your brother's room, which one is going to go to the grandparents' room, like that. First of all, what you are doing? You are sorting. After you sort, what you are doing? You are doing modification according to their preference, right? So sorting, modification, and then what you are doing? You are labeling. This one is going to go to your room, this one is going to go to their brother's room, this is going to go to your parents' room, this is going to go to your grandparents' room. So modification, sorting, labeling, right? And then you cannot just send it like that. You probably also have to package it properly so that it does not drop or it does not do anything. So all these things are happening in the what? Golgi apparatus. So after you do all the modification in the Golgi apparatus, now you want to send it to the protein where they are gonna try to go, right? That is also happening by the what? The help of the transport vesicle. That is also happening by the help of the what? Transport vesicle. Okay, so now everything is done. Information is here in the nucleus. cooking is happening in the ribosome and endoplasmic reticulum. Endoplasmic reticulum also helps with the modification, and from there you are sending with the transport vesicle the protein to the Golgi apparatus to be able to do labeling, sorting, modification and packaging, and then you are send like dispatching to the what specific place, right? That is also happening by the transport vesicle. So now whenever you were trying to do all these on the dining table, did ever that happen that some tacos did not come out well? So these are not useful. Nobody's going to eat that. What are you going to do with that taco? You probably want to recycle it, right? Who's going to recycle it? So you have another type of the organelle called lysosome. What do you have? Lysosome. What do you have? Lysosome. Lysosome is also called the what? Suicidal sac. Do you know why they are called the suicidal sac? Because they have the digestive enzyme to break down the protein. So protein is made up of the what? Amino acid. Remember I told you monomer of the protein is what? Amino acid. Ingredients are what? Amino acid. You cannot probably use that taco, but you probably can use that ingredient to make another dish, right? So that is happening in the what? That is happening in where? In the lysosome. Okay. So now if anything bad is there, like for example, some microorganism is there, some pathogen is there, or maybe while you are trying to make a lot of generating ATP, you are making some toxic molecule like hydrogen peroxide. So the hydrogen peroxide is toxic for your body. So what do you want to do? You want to detoxify it. That detoxification is happening in the what? Peroxism. That detoxification is happening where? In the peroxism by the enzyme called hydrogen peroxidase and catalase. Okay. So by using the peroxidase, what you are doing? You are converting the hydrogen peroxide. First you are making the hydrogen peroxide, then from there you are making the what? Water molecule and the oxygen, so that this water and oxygen, both of them are not toxic to your body, but hydrogen peroxide is what? Toxic to your body. Okay. So this is exactly what is happening inside your cell. So now I'm going to go over each slide very quickly, because you know the story already, what's going on. Okay. I'm not going to spend a lot of time. So now everyone knows what's happening in the mitochondria. What's happening in the mitochondria? You are generating ATP, and mitochondria is called the powerhouse of the cell. And another thing about the mitochondria is mitochondria also has their own DNA, RNA and ribosome. The reason they have their own DNA, RNA and ribosome is because once upon a time mitochondria was a what? Bacteria. Okay. So that bacteria all of a sudden decided to stay in a community, right? After staying in a community, they are like, okay, I'm gonna generate ATP for you, you're gonna provide all other facilities to me. Okay. So that is how they started becoming the part of the house, like the cell. Okay. And then the ribosome. So what do we mean by the ribosome? Ribosome is the what? Kitchen. And ribosome is made up of the what? Ribosomal RNA. Why ribosome is called the kitchen? Because ribosome allows what? What happens in the ribosome? Protein synthesis. What happens in the ribosome? Protein synthesis. So you have two different type of the ribosome. Some of the ribosome which are attached to the endoplasmic reticulum, we call them membrane bound ribosome, and some ribosomes are what? Free. We call them free ribosome. The free ribosome are free floating site of the synthesis of soluble protein that function in the cytosol or other organelle. So these are the free, they are making the soluble protein which can be used inside the cell. But this one is making the protein which can go to so many different other places, right? So the membrane bound ribosome attached to the membrane of the endoplasmic reticulum, site of the synthesis of protein to be incorporated into the membrane or lysosome or exported from the cell. Then you have the endoplasmic reticulum. So you have the rough endoplasmic reticulum right here, which is the site of the protein synthesis, but the smooth endoplasmic reticulum right here, which does not have ribosome on top, is not associated with the protein synthesis. Instead, this is associated with the lipid metabolism, fat transportation, detoxification, glycogen to glucose conversion, calcium storage. Very different, okay? So rough endoplasmic reticulum associated with the protein synthesis and the modification, and your smooth endoplasmic reticulum does not do anything associated with the protein. They do what? Lipid metabolism, fat transportation, detoxification, glycogen to glucose conversion, and the calcium storage. So now what is happening in the Golgi apparatus? Who remember the dining table in the Golgi apparatus? What you were doing? You were labeling, sorting, labeling, packaging, modifying, and what? Packaging, right? Transport vesicle from the endoplasmic reticulum fused with the cis phase of the Golgi. Proteins or lipid taken inside the further modified, tagged, sorted, and the package. And Golgi is traffic director, controlling which of the three pathways final products will take as new transport vesicle pinch of transport transphase. So exactly what I told you all about. You are making the protein in the rough endoplasmic reticulum with the transport vesicle. You are sending it to the Golgi apparatus. In the Golgi apparatus, you are sorting, packaging, labeling, modifying, and then dispatch it with the transport vesicle to their designated place, right? Okay, so the next one is what? Peroxism. So everyone knows what's happened in the what? Peroxism. In your cell, while you are generating ATP and doing all this function, you are generating some toxic molecule. So to detoxify those, you have some enzyme called the oxidase and the catalase, so that you can actually detoxify the toxic molecules. And who remembers about the lysosome? Lysosome has the what? Digestive enzyme to be able to what? Break down the protein which you do not use and recycle it. Recycle the ingredients. So now when we say endomembrane system, all these organelles together is called the endomembrane system, because together what are they doing? Together they're producing, degrading, storing, and exporting biological molecule and degrade potentially harmful substances. Now let's talk about the cytoskeleton, which actually provide the what? Which actually provide the structure of the cell. So you have three different types of them: microfilament, intermediate filament, and the microtubule. Let me focus on the microtubule, but that is the most important one. So when we say the microtubule, so a lot of the time cell can have a finger-like projection, and we call them what? Villi, microvilli. Okay, usually you see the microvilli in your intestine to increase the surface area so that they can absorb the nutrient very efficiently, right? And sometimes you can have flagella. Flagella is for moving. Okay, the reason you can hold this structure is because of the presence of the what? Because of the presence of the cytoskeleton, microtubule, the structure, right? And sometimes also, like for example, in your respiratory tract, you can have some hair like structure to move things through. Okay, these are called the cilia. Flagella is for the movement of the cell, microvilli is for the absorption of the cell, and these are happening because of the what? Structure of the which structure? Because of which structure? Because of the structural component, microtubule. And also the microtubule together make the centriole, and two centriole together is called the centrosome. So whenever I talk about the cell division, I will talk about how this centriole and centrosome actually help you to what? Help you to divide the cell. Okay, so what is the function of the centriole and the centrosome? cell division. And then what is the function of the microtubule? Microtubule together is forming the what? Centriole and the centrosome, and they also can help forming the what? Microvilli, cilia, flagella, all this. Yeah, cilia and flagella, I talked about it already. I talked about the microvilli as well. So finger-like projection to increase the surface area. Okay, so now let's see how much you remember. Which organelle is involved in the energy production? Mitochondria, very good. Which membrane-bound organelle function primarily in digestion within the cell? Lysosome, very good. What is the function of the Golgi apparatus? Sorting, labeling, modifying, and packaging and dispatching. So what structure of the cell's external surface is found in parts of the respiratory tract? In the respiratory tract, cilia to move things around and participate in moving mucus toward the throat. Yeah. Okay. So this is something nucleus, I already talked about it, but nucleus has some structure. So in the nucleus you have the nucleolus, and this is the what? Chromatin, and these are the what? Nuclear membrane. Okay, and here in the nuclear membrane you see the what? Nuclear pore. Why do they have nuclear pore? So that when you are making the RNA from DNA, the RNA can actually what? Go out of the nucleus. That is why you have the what? Nuclear pore. So you have the nuclear pore, you have the nucleolus, which is the site of the ribosomal assembly, and most of the cell in our body is actually uninucleate. Okay, that means at least you have one nucleus in the cell, but some cells can have multiple nucleus. Okay, but red blood cell does not have any nucleus. Okay, so now let's talk about some important understanding. We only know that this part right here that you see, it has what? DNA. Not only it has DNA, it also has the what? Protein. DNA and protein together is called the what? Chromatin. What do you call it? Chromatin. DNA and protein together is called the what? Chromatin. Actually, let me tell you another analogy, then you will understand. So have you ever seen like long thread? Yeah. So if you have a thread that is very long, what do? Huh? You wind it up around a ball or something, right? Exactly like that, you have a very long stretch of DNA. So to fit the what? DNA properly inside your nucleus, what do you have to do? You have to wrap it around the protein. We call those protein histone protein. What do we call it? Histone protein. The histone protein and the DNA together is making the what? Chromatin. Okay, chromatin. What are they making? Chromatin. Now, whenever your cell is ready for the cell division, that's when you make the what? Chromosome. So your chromatin actually absorb the water. They actually what? Become thick and visible under the microscope, and they exactly look like this. And this is called the what? Chromosome. So now let's talk about the cell division. Cell division is basically the part of your next lab. So now let's talk about it. So what is happening during the cell division? So before that, let me just ask you some question. Do you know you all came from one cell? Yeah, what is the name of that cell? Zygote, very good. So now, do you know that you are a body of actually trillions of cells now? Yeah, so how did that happen? So your cell actually divided, right? One cell to two, two to four, four to eight, like that. Your cell has actually what? Divided. So your cell, all of your cell has a cycle. All of your cell has a what? Cycle. So the first your cell is born, and then your cell has to actually grow and carry on its the usual activities, right? And that is called the preparation phase or the interphase. What do you call it? Preparation phase or the interphase. So during the preparation phase or the interphase actually has the three different phases. Is called the growth 1 phase, synthesis phase, and the growth 2 phase. So in the growth 1 phase, what is basically happening is your cell grow. And then synthesis phase. What is the most important part of your cell? What is the most important part of your cell? DNA. So the recipe book. So you want to make sure if you do not have the recipe, you cannot cook food. You cannot make protein. You cannot survive. So that is the most important part. So before you actually what? Before you actually divide the cell itself, you want to make sure you are copying the information from one recipe book to another recipe book. What is happening? So you are basically duplicating the DNA or replicating the DNA. So the DNA replication happened during the synthesis phase. Okay. After the DNA replication happens, so you have the 2 copies of the DNA. What do you have? 2 copies of the DNA. These 2 copies of the DNA is called the what? These 2 copies of the DNA together is making the chromosome. These 2 copies, one copy right here, another copy right here, these are called the sister chromatid. What do you call it? Sister chromatid. These sister chromatid are actually attached to each other by the what? Centromere. They are attached to each other by the centromere. So when you do the cell division, what you are basically trying to do, you are trying to separate 2 sister chromatids so that each of the recipe book can go to the each new cell. Does it make sense? So very important is before you do the cell division, you do the what? DNA replication, and DNA replication happens during the what? Synthesis phase. So then after the synthesis phase, what is happening? Growth and the final preparation, right? And then they are getting ready for the cell division. So that cell division is happening by the 2 steps. Number 1 is the nuclear division. Nuclear division, in the nuclear division, what you are basically doing is you are separating these 2 what? Sister chromatid. Okay. So prophase, metaphase, anaphase and telophase. These are the steps to separate this what? Nucleus. And after you separate the nucleus, what do? You separate the cytoplasm by the cytokinesis. So in the cell cycle, you have the growth 1 phase and the synthesis phase and the growth 2 phase, and then prophase, metaphase, anaphase, telophase and the cytokinesis. So now let's talk about the DNA, right? If you look at the structure of the DNA, if I start, I can talk about it forever, but I want to just let's see how much I can simplify. So you have the, if you look at the structure of the DNA, you have the what? 2 strand of the DNA. And usually, remember I told you, like DNA, the monomer is called the nucleotide. In the nucleotide, you have a sugar molecule, you have a phosphate molecule, and you also have a base. Okay. So now, for example, if I want to draw the DNA, like for example, the sugar and then phosphate and the base, sugar, phosphate, 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 base. So the base could be either adenine, guanine, cytosine, or thymine, adenine, guanine, something like that. Okay, this is your one strand. Another strand is actually opposite. So they have the phosphate here. Okay, the phosphate here, the phosphate here. So what does that mean? It means that you have the 2 strand. These 2 strands are actually what? Anti-parallel. One is going this way, another is coming this way. Okay, they are what? Anti-parallel, and they are also what? Complementary. When I say complementary, what does it mean? It means A binds with T. So if you have A in one strand, you have T in the other strand. If you have G in this strand, you have C in this strand. If you have C, you have G in this strand. If you have T, A in this strand. If you have A, you have T in this strand. So that is why your DNA is double stranded anti-parallel and what? And complementary to each other. Okay. So now whenever you are doing the DNA replication, by the way, when DNA replication happens? DNA replication happens during the synthesis phase, during the synthesis phase. So whenever DNA replication happens, you are basically copying the information from one DNA to another DNA. So for example, you have 1 and 2, so 2 strand, so you 1 strand, 2 strand, you are making, so for example this is 5 prime to 3 prime, so 3 prime right here, and this is the opposite, 5 prime right here and 3 prime right here. So when you are replicating the DNA, the replication of the DNA is happening by the enzyme called DNA polymerase 3. So the DNA polymerase 3 can make the new DNA strand only in one direction, which is the 5 prime to 3 prime direction. So that is why you have this DNA replication. In one strand they can make the DNA at one stretch, but whenever you are making the DNA in the other strand, you have to make it as a fragment. That is, these fragments are called the Okazaki's fragment. Okay. First of all, what is the process of DNA replication? First of all, your DNA is what? Helical. So you have to first make it straight. Who helped to make it straight? The enzyme called topoisomerase. And there is an enzyme called helicase. Helicase is the one which actually what? Separate the strand of DNA by removing the hydrogen bond. Okay. And then you have the topoisomerase to unwind, helicase to what? Separate the 2 strand, and what? DNA polymerase 3 to make the new strand of the DNA. And this process is called the semi-conservative DNA replication, because you are using one old strand and you are making the what? Another new strand. That is why the process is called the semi-conservative DNA replication. I don't know, probably I talked about it in like 2-3 minutes, but I can talk about the DNA replication for 2 hours. There are so much of details that are happening. Yeah. Now I talked about all of this, the DNA polymerase, semi-conservative replication, all of this, right? Probably I did I talk about the DNA ligase? So whenever you are making the short fragments of the DNA, you have to stitch them together by the enzyme called DNA ligase. Okay. So as you can see right here, first of all, you have the helical double-stranded DNA. Topoisomerase is making them straight, and helicase is separating them, and the DNA polymerase is making the new strand of the DNA. So what is the name of the enzyme that separates the strand of the DNA during the DNA replication? Helicase, very good. In which phase does DNA synthesis occur? Synthesis phase. Where does this occur in the cell? DNA replication happens in the nucleus. Why is DNA replication called the semi-conservative? Because you have one old strand and then you have another new strand produced. Now when you look at the cell cycle one more time, this is the interphase synthesis phase. You are doing the DNA replication. After you do the DNA replication, you are going to divide the cell. To divide the cell, first of all, what you want to do? You want to separate the sister chromatid by the process called mitosis. And there are 4 stages: prophase, metaphase, anaphase, and telophase. So now let's talk about this very important one. So when we talk about the interphase, during the interphase, your nucleus, in the nucleus, your chromatin is in the chromatin stage. This is the chromatin stage. It looks like the what? Thread, right? Thread with what? Wrapped around the histone protein. This is called the chromatin. So whenever you cannot see the chromosome clearly under the microscope, you can tell this is what? Interphase. Okay. So when they start the cell division, the first thing they are gonna do is they are going to absorb the what? They are going to absorb the water and become what? Short and thick and visible under the microscope and form the what? Chromosome. So as you can see, the chromosomes are very clearly visible under the microscope. And what happens? The nuclear membrane starts disappearing. Okay. And then remember, there is an organelle called centriole. This centriole is the one which is going to generate the mitotic spindle. What they are going to generate? Mitotic spindle. So in the mitotic spindle, you have all this, you have mitotic spindle. These are all like microtubule. So in the microtubule of the mitotic spindle, the centromere of the chromosome is going to get attached and we call it kinetochore. So that is exactly what is happening during the prophase. So in the metaphase, all of this chromosome is going to be what? Aligned in the middle of the what? Cell. In the mitotic spindle, we call it metaphase. And in the anaphase, these two sister chromatid will be pulled to the polar region. We call it anaphase. And in the telophase, they are going to reach the what? They are going to reach the tail, reach the end. Okay. And then the cytokinosis will start to happen by producing the what? Cleavage furrow right here. And they are going to separate the what? You have already separated the nucleus. Now you are separating the what? Cytokinesis. So in your lab, you should be able to actually what? Identify the different stages of the cell cycle. We are going to talk about it and we are going to do some practice. So in the cytokinesis, what is happening? In the mitosis, you divided the nucleus. Now in the cytokinesis, you are dividing the what? Cytoplasm. Very good. So now let's talk about protein synthesis. So we have learned about the DNA replication which is happening in the nucleus. Now for the protein synthesis, what are the steps? Do you remember? First you have the recipe book. From the recipe book, you copy the information to the paper. So what does that mean? You are copying the information from DNA to RNA, and the RNA that you are producing, we call it messenger RNA. The synthesis of the messenger RNA is actually called the what? DNA transcription. What do you call it? Transcription. After the transcription, that messenger RNA comes out of the what? Comes out of the nucleus, and then transfer RNA from the store bring the ingredient to the ribosome. And then what do? Based on the information that you have, you start making the what? Cooking the protein, right? That is called the translation. What do you call it? Translation. So DNA to RNA transcription, RNA to protein translation. So now you have three different type of the RNA. I told you, right? Messenger RNA. Messenger RNA is the RNA that you are making from the what? DNA to RNA, right? And transfer RNA is the one which is going to bring the ingredient by reading the what? Ingredient list. And the transfer RNA and ribosomal RNA is basically the structural component of the ribosome. So now we know about the messenger RNA already. So the messenger RNA has the what? So now let's talk about the whole entire thing right here. So what is happening? So you have the DNA. DNA have the information to make the protein, right? What is the information that you have to make the protein? So we know that these are the bases, right? TAC, like three bases together is actually information for one ingredient. That is why they are called the triplet, or they are also called the what? Codon. Okay. So you are actually, what you are doing? You are copying the information from the DNA to messenger RNA, right? You are copying the information from the DNA to messenger RNA. Now the ribosomal RNA, by reading this is the codon, and transfer RNA actually read it backward, and we call it anticodon. Okay, and bring the ingredient by following the information. TAC or AUG is responsible for bringing the what? Methionine, that is the start codon. right? So that's where you start. You start codon, you bring the methionine. That is where you start the protein synthesis. So there is also information to stop the protein synthesis. That is called the what? Stop codon. So now for the transcription, there could be very elaborate answer, but I'm just going to tell you in a very short. Like there are three processes. First of all is called the what? Initiation. In the initiation, before the gene or DNA, you have a specific place called the promoter. In the promoter, RNA polymerase, which is responsible for generating the new messenger RNA, is going to get attached, and then elongation. By reading whatever information you have, you are generating the messenger RNA, and then by the termination, when you get the what? Stop codon, you stop the what? Generating the new messenger RNA. So after you stop that, so for example, you have copied the recipe book, right? From the taco recipe, for example, you have copied the information. So now you have different codon, okay? Now let me tell you, so a lot of the time when you look at the recipe book, some of the things probably, some of the ingredients probably do not like, right? Do you copy exactly the same? No, right? Sometimes you remove some of the ingredient which probably you do not understand what is this, what they're gonna do, things like that. So you just remove all those part. So those part of the DNA is called the what? Intron. What do you call it? Intron. You only keep the information which is called the what? Axon. What do you do? You only keep the information which is in the axon. Okay, so when you are removing the intron from the messenger RNA, that is called the RNA processing, or this is also called the what? Splicing, happening by the protein called spliceosome. Okay, the function of the spliceosome is to what? Keep only the ingredients that you want to keep and remove the one which you do not want to keep. Okay, and we know that from the DNA you are making the RNA inside the nucleus, and that RNA should get out of the nucleus. Nucleus is a very safe place, but when they go out, they could get damaged. So that is why also you add a 5' cap and a poly A tail, so that they do not get degraded outside of the nucleus. So now you have all the genetic information. Based on the genetic information, transfer RNA is going to bring all the amino acid and put them together to make the what? Protein. So like initiation, elongation, and the termination when they reach the stop codon. Sometimes a lot of our ribosome together can make one polypeptide. Okay, I know I'm going like so fast. So you already made the protein. After you made the protein, what do you do? Who remembers? After you make the protein, what do you do? Nobody remembers anything because I'm so fast, right? Hmm? In the endoplasmic reticulum, right, you are what they have the signal sequence on a growing polypeptide chain. Signal associated the ribosome to dock on the rough endoplasmic reticulum surface, and then the signal recognition peptide on the endoplasmic reticulum directs the mRNA ribosome complex where to dock, where they're gonna go. Okay, they are putting the signal over there, and once docked, forming polypeptide enters endoplasmic reticulum. Sugar groups may be added to the protein and its shape may be altered, and protein is then enclosed in the vesicle for the transport to the Golgi apparatus. Remember, does it make sense when I talked about the story? From the kitchen, transport it to the where? In the dining table, from the dining table to the specific rooms. Okay. So far we have talked about the RNA, which is responsible for the gene expression to make the protein. But there are some RNA which actually want you to stop making those protein. So there are some protein which could be actually harmful for you. So your body can actually has the way to stop producing them. Okay. For example, in your home, you cook taco. Probably taco is probably a good food, delicious and healthy food. But what if you want to always make some fried food? Is it good for you? Not good for you. So probably your mom wants to stop making it. So that is happening by this one, microRNA, riboswitches, small interfering RNA. They do not want you to cook that specific protein, okay, which is probably bad for you. Okay. So now autophagy. Have you ever heard of the term autophagy before? No, autophagy. So remember the lysosome that you have, autolysis, they have the what? They have the digestive enzyme to degrade anything that is bad for you. So there could be some protein which are bad for you. There could be some organelle which is not working properly. What they are gonna do? Lysosome has the digestive enzyme to break them so that you can recycle the ingredient to use it for something else. Okay, that is happening by the autophagy. Self-eating is the process of disposing of the non-functional organelles and sweeping up the cytoplasmic bits by forming autophagosome, which can then be degraded by the lysosome. Now, cell cleanup pathway. So for example, a lot of the protein that I told you, a lot of the tacos that you made probably did not come out good. So nobody's going to eat that. What are you going to do? You're going to tell them these are not good, bad. So your cell actually put some protein for the destruction. We call them ubiquitin. When they attach to the ubiquitin, this is not good anymore. You want to destroy it. So to destroy it, you have the proteasome disassemble ubiquitin tag protein and recycling the amino acid and the ubiquitin. So this is for destruction. So you just recycle it. Okay. Then the last, we are almost done. Apoptosis. So not only the organelle that is not good, you want to destroy it. What if the cell is not good? What do you want to do? You kill them. Programmed cell death. You kill the cell, which is actually good for you. A lot of the cells sometimes get bad, so you want to kill them. That is the process called the apoptosis, also known as the programmed cell death. Causes certain cells, example cancer cell, infected cell, and all cells to neatly self-destruct. Process begins with the mitochondrial membrane leaking the chemicals that activate an enzyme called caspases, and caspases causes degradation of the DNA and cytoskeleton, which leads to the cell death. And that cell shrink and is phagocytized by the macrophages. So after you are done with the death cell could be what? Eaten by the macrophages. Okay, then cell aging. So we have been talking about the cell is, you have a cell, the cell is growing. At the end, you want to stop them dividing, right? So it depends on actually what? Every time you divide the cell, the end of the cell is called every time you divide the DNA, the end of the DNA is called the what? Telomere. What do you call the end of the DNA? Telomere. So every time you divide or replicate the DNA, the telomere length shorten. So it shortens at a point where you cannot replicate the DNA anymore. That is the point you actually stop dividing the cell. So the telomeres are the string of the nucleotide that protect the ends of the chromosome. It's almost like the shoestring, right? And then the what? Caps on the shoestring. And every time a cell divides, the telomere shortens, so the telomere may act like an hourglass on how many times a cell can divide. But you also have an enzyme called telomerase, which can actually lengthen the what? Telomere. So modified rates of the cell division. So sometimes you can actually increase the cell division, right? So for example, have you ever heard of like the more you use your muscle, the more muscle you grow, right? So we call them what? Hypertrophy. But if you have seen after the paralysis, what happens? You are not using that part of the muscle and then you start losing that muscle, right? That is called atrophy. So you can actually increase the cell division or you can actually decrease the cell division. When you decrease it, you call it atrophy. When you increase it, you call it hypertrophy. And also hyperplasia is accelerated growth that increases the cell number when needed. And then all the processes that we have been talking about, the protein synthesis, the protein, DNA, all this, if you have mutation in the DNA, you will have the bad protein, right? So for example, one of the diseases is called the what? Progeria is a rare disease that mimics the aging. So it is caused by defective progerin protein. So what does it mean? Probably your DNA did not have the proper recipe to make the proper progerin protein. Due to that, you are making a defective progerin protein, which will make you look very, very aged. Okay. So disease appears by age 2. Children with diseases display slow growth, thinning hair, brittle bones, arthritis, severe cardiovascular disease, with death usually by age 20. Scientists have found drugs that stimulate the autophagy that can help clear that bad progerin. Okay, this is the last slide. We are on time. So what is the codon and anticodon? Codon is the triplet code that you have on the messenger RNA. And anticodon is what? Your transfer RNA read that codon in what? Complementary way. And that is called the anticodon. Where does the transcription take place in the cell? And translation. Transcription happens in the nucleus and translation happens in the ribosome, the kitchen, protein synthesis. What initiate the transcription and translation? Remember that switch, you turn on the switch and transcription happen and then the translation happen. For the transcription to happen, promoter has to bind with the what? Promoter has to bind with which part? DNA polymerase has to bind with the promoter, and for the translation to happen also something like that. So what terminates the translation? When they reach the stop codon, they stop the translation. Compare processing of mRNA with the protein. So whenever we are talking about the mRNA processing, what you are doing? You are doing the splicing to what? Remove the intron and keeping the what? Axon, and then putting poly A tail and the 5' cap. But whenever we are talking about the processing of the protein, what we are doing? We are doing modification, right? Adding carbohydrate or adding lipid or all these things. Okay, that's it. That's all about this chapter. Does anybody have any question? No question. I know you guys are